Many of you will know that we have our annual awards competition, and just heard this afternoon that we've had 170 applications for this year's competition. So it looks like being a lively activity again. And it's our custom, as many of you will be aware, to ask the award winners to come and give us a glimpse of their work. And this evening I should say, Hugo Poplimont, who's going to talk about how neutrophils organize themselves to stop inflammation. Hugo from Vienna, please speak to us.
Hello, can you hear me? Hello, yes, so you can hear me, right? Good. So yes, thank you very much for inviting me. I'm very happy to talk to you all tonight. It's a great opportunity for me to present the work I did during my PhD. So as you mentioned, I studied how neutrophils organize their migration to sites of inflammation in vivo. And I did that in the laboratory of milk assays in Cambridge. And the results of this work are published in Current Biology. So why it's important to understand how neutrophils organize their migration? So it's important because it's very important to understand how inflammation works. So inflammation is initiated by immune cells, and it's a process that is very important actually, and that can be beneficial because it's the process used to clear microbes from your organism, but it's also a process that can be detrimental if there is too much inflammation, like chronic inflammation that can lead to stroke, heart disorder, respiratory disease. And so I'm focusing on neutrophils, those small immune cells, because actually they are the most abundant immune cell type in the human blood, and are among the first immune cells to reach site of inflammation. So first they are in the blood, and then they start adhering to the endothelial cells from the circulation, and then they get into the target tissue. And actually I was interested in how they get to the site of inflammation. So it could be an injury, wound, or an infection. So I'm interested in how do they get there. So what we knew so far was that they use an attractant that is called LTB4, leukotriene B4, and that's how they sense the inflammation. So they can produce it actually. And so we wanted to know actually how all these mechanisms work, and how do they have this coordinated migration to site of inflammation, how this inflammation starts basically. So what are the cellular events that are leading to the production of this key attractant that is LTB4, leukotriene B4. So to do that, I used the zebrafish model. So this is a zebrafish. So we use the zebrafish larvae. You can see that they develop pretty quickly. And so in this larva that is actually around 3 days post-fertilization here at the end, there's already a circulation, there are already neutrophils, there are already macrophages. And so it's a very good model also because the larva is transparent. So I can genetically modify the larva and make them express fluorescent protein. Those fluorescent proteins are going to be expressed only in the neutrophils, so I can track the neutrophil and take videos of those neutrophils going to sites of injury or sites of infections. So this is an example. So here you have the zebrafish, you can see, and I'm going to show you how I did a laser wound on those zebrafish. So basically, this zebrafish has been genetically modified, so it expresses a fluorescent protein in those immune cells that are the neutrophil. And I'm performing a wound with the laser of the microscope. And so I'm triggering the inflammation process. And so the neutrophil are going to migrate to the wound. So you can see the neutrophil in green. And this is the site of the wound. And all the neutrophils are migrating. And their migration is quite coordinated. And then they stop at the wound where they form this cluster. So the thing is, this GFP, it allows us to follow the neutrophil migration, but we don't really know what's going on. What are those neutrophils doing when they are producing the attractant? So we needed a better sensor. We needed better tools to study this. So how can we monitor the events leading to the attractant biosynthesis? So I'm going to skip a little bit on this because it's a bit complicated. But basically what you need to know is, in order to produce this attractant that is very important, LTB4, that is here, some enzymes that are present in the nucleus needs to move to the membrane of the nucleus. And usually they do that after an influx of calcium into the cell. So when the intracellular calcium concentration in the cell increase, those enzymes get activated and they migrate to the membrane of the nucleus, where they start producing molecules that will lead to, and this will lead to the production of the molecule of interest, LTB4. So basically what I did is I genetically modified the fish for them to express a calcium probe. So this calcium probe actually is getting brighter as the calcium concentration increase into those immune cells. And here you can see a video very similar to the one I showed you before, except that the neutrophils here are expressing this calcium probe. So the highest the fluorescence is, the higher the calcium concentration is in the cell. And you can see that neutrophils are in the cluster, they have high calcium. So probably that's those neutrophils are producing the attractant we are interested in. But it's not really sure. So to be even more precise, what I did is I attached a fluorescent protein to this enzyme that is very important for the production of LTB4 or key attractant. And so with this fluorescent protein attached to the enzyme, I can actually follow the enzyme and I can see when this enzyme get activated, because I can see when this enzyme get translocated from the nucleus to the membrane of the nucleus. And so again, I perform a wound on those poor zebrafish. And on the left, you can see the same cells expressing GCAMP, so the calcium probe. And here you can see cells expressing this probe. And you can see here, actually on this corner, there was a zoom. I'm going to play it again. And you can see here that the enzyme is translocated from everywhere to the nucleus to the membrane. It's making those donut-shaped nucleus. And so with this, I was able to determine that those neutrophils are producing this key attractant when they are at the cluster and when they have this very high intracellular calcium concentration. It was still a mystery what was propagating this signal, what was propagating the signal that was telling the neutrophil to start producing this attractant. So what was the signal that was making the neutrophil, making the intracellular calcium concentration in the neutrophil increase? And actually what we realized is that first neutrophil, they can sense the dead cells that are at the wound, and they sense the ATP in those dead cells, and what they do is like they exchange molecule of ATP through connexins ME channels, and those molecule of ATP are going to bind another receptor that let actually the calcium in. And when neutrophils touch each other, they can basically transmit those molecules of ATP, open up the channel, and there is an increase of intracellular calcium concentration, and this triggers the cascade that will lead to the production of this key attractant. So basically, that's why everything is happening in the cluster. And this is very new, because before people thought that they were producing the attractant as they were moving, but actually, no, they need to stop, they need to stay there, they need to have very high intracellular calcium concentration, and they make those big cluster, those factory of inflammatory molecules that will trigger the recruitment of many immune cells. So to prove that, actually, I inhibited those channels that actually let ATP go, so the connexins channel, by using, for example, carbonexolone, a drug, and also by genetically inhibiting this gene in the fish. And you can see here, actually, those neutrophils that are supposed to be very bright and that are supposed to cluster at the center where the wound is. So the wound is here. You can see the dead cell of the wound actually stained in red here, because I stained them with propylidomide. But you can see that the behavior is totally different. And neutrophils are actually just an exploratory behavior. So we were able to actually determine that those connexin channels were crucial for the production of chemoattractant, and were crucial to control the amount of neutrophils that were going to the site of inflammation. But we wanted to know more, And so I designed an assay before leaving the lab. And this new assay actually is to monitor the infection of a wound by a pathogen. So I use Pseudomonas aeruginosa, which is a bacteria that is really, really good at contaminating wound. And so basically I leave the fish in a bath of Pseudomonas, and then I trigger a laser wound on the fish. And so you can see in live the bacteria invading the wound. And you can see the response by the neutrophil. And so that's what you can see here. So you have the bacteria in red, you have the laser wound here, and the neutrophil are in green here. And you can see that the bacteria are going to come because they sense the wound as well. And they come and they want to enter the body of the fish. And then there is the response from the neutrophil. And I performed the same experiments with those inhibitors of those channels, the connexin channel that I talked to you about. And actually, by inhibiting the connection channel, the neutrophils were not able to reach the wound and cluster there. And they were not able to actually prevent the infection. And the mortality in the fish was increasing by using those inhibitors. So the takeaway message is that those connexin heavy channels are very important, and they appear to modulate the local response to injury and to pathogens. And they are crucial to control basically inflammation. And I think it's very important right now to control inflammation as there is more and more antibiotic resistance. We need to find new ways to fight pathogen, and this way could be, for example, to enhance the immune response specifically at the site of infection, for example. So I'd like to thank everyone in my lab, the fish facility, and of course, my PI, Milka Saris. And just a last slide to introduce you what I'm doing right now. So in Vienna, so right now I just moved in Vienna, crazy time. And right now what I'm doing is I'm grafting cancer cells into zebrafish. I love this model, so I stayed with this model. So I'm staying with the zebrafish larva, but I'm grafting cancer cell. And what I'm doing is actually I'm using thousands and thousands of larvae, and I'm testing a lot of drugs on them. And basically, I can automatically test drugs on those fish. And so I'm trying to find drugs that inhibit the spreading of cancer cells. So what I do is I graft the fish with cancer cells, and then I look at how cancer cells spread. So here you can see cancer cells in green that are spreading across the body. And basically, I'm trying to find a drug that will inhibit the spread of cancer cells. That's what I'm doing right now. So thank you very much for your attention. And if you have any questions, don't hesitate to ask me.
Well, Hugo, that was quite remarkable. Thank you very much indeed. And as you say, what a critical kind of response this is, inflammation, and it affects all sorts of things, different diseases, and of course all of us get it at some time. Can I just remind people, I think we've got time for just one or two questions for Hugo, but can I remind you, the old lags know the routine. If you would like to ask a question, please do not use the chat function because that gets a bit complicated. There are two options. One is to raise your hand, in which case Valerie Anderson, who is behind the scenes, will spot you and try and bring you in if you wish to make a live contribution. If on the other hand, you would like to just write in a question on the Q&A, that's fine too. And what I will then do is endeavor to very lightly edit the contributions and read out what the question you would like asked. So if, like me, you're rather shy at being on the telly, just send us a written question into Q&A. So Hugo, perhaps you could shut down your slides if you wouldn't mind.
Yes.
Thank you.
Sharing.
Yeah. And then are there any questions that people would ask? I'm not seeing anything on the Q&A. Valerie, do we have anybody with their hands up? Okay. So there's just, I can just see one hand, which is John Cook. So if you'd be kind enough to ask your question.
Thank you, Hugo. That was fascinating. the biochemistry is a little over my head, but I was fascinated by the experimental techniques that you used and the microscopic images. So I presume this is optical confocal microscopy that you're using, is that right?
Indeed, I'm using a mix of confocal microscopes. I'm using the spinning disc, classical confocal microscope, but for the laser wound, actually, I'm using 2-photon microscopy. Okay. Because you need the 2-photon microscope to be able to perform laser wound at such high intensity. Okay. In order to properly wound the fish. At
the top left of your images, there was a time scale, and which was running from 0 to 30, 40 something. Are those seconds?
There are minutes. There are minutes.
Oh, they're minutes. So significant. Yes. Those were significantly speeded up.
Yes. Okay. So the cells move approximately. So the cells are approximately 10 to 15 micron big, and they move. Full speed is really like 15 micron per minute.
Okay.
Approximately. So the size of a cell per minute.
Well, that's still
astonishing.
They have to squeeze their body into this complex environment.
Great. Thank you, John. Thank you, Hugo. That was absolutely fabulous. I think I'm going to stop the questions there and move on. But thank you, Hugo. Again, good luck. And I feel a bit sorry for the poor old fish, but the benefits vastly outweigh the trials of the fish, I suspect. So thank you again for that, and enjoy Vienna.
Thank you very much.